Well, good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled Bispecific Antibodies, Structural Design, Production, and Purification Strategies. My name is Samaya and it's my pleasure to be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll attend to your questions during the Q&A. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, you can contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Genscript who developed the content for this presentation. Genscript is the leading contract research organization in the world providing gene, peptide, protein, CRISPR, and antibody. Since its foundation in 2002, Genscript has grown exponentially through partnerships with scientists conducting fundamental life science research, translational biomedical research, and early-stage pharmaceutical development. The company is recognized as having built a best-in-class capacity and capability for biological research services encompassing gene synthesis, peptide synthesis, custom antibody and protein engineering, and in vitro and in vivo pharmacology. Now it's my pleasure to introduce our speaker for today's event, and our speaker is Dr. Xiao Chen. Dr. Xiao Chen obtained her science PhD from the Center of Excellence in Molecular Cell Science, Chinese Academy of Sciences. Her main project involves investigating the structure and function of G protein coupled receptors, leading the analysis of the first alpha-2C adrenergic receptor protein structure. The research finding was published in Cell Reports. So now, without further ado, I'd like to pass control to our speaker. So whenever you're ready, you can go ahead and get started.
Hello everyone, thank you all for joining this webinar today. My name is Xiaoyu Chen, Senior Scientist from Genscript. I'm responsible for Bispecific Antibody Expression Service, and I provide support to Bispecific Antibody design, purification, and troubleshooting. The topic today is Bispecific Antibodies, structural design, production, and purification strategies. As you may know, Genscript has been dedicated on recombinant protein expression for more than 19 years, and already delivered over 100,000 projects. Also, we have done thousands of bispecific antibodies. I'm very glad to share with you our experience on bispecific antibody design and production. I'll briefly introduce bispecific antibody, and then challenges we'll meet when designing and producing bispecific antibody. And finally, I'll show Genscript experience on bispecific antibody production. First of all, what is bispecific antibodies? Bispecific antibodies, or BsAbs for short, have two distinct binding domains that can bind to two antigens or two epitopes of the same antigen simultaneously. However, random assembly of two different heavy chain and two different light chain results in 10 different molecular configurations, among which only 12.5% represent the desired BsAb statistically. That's why the development of BsAb was slow in the first 3 decades, until some important engineering strategies was investigated, such as knob-in-hole, cross-map, dual-body. For now, there are over 100 different BsAb formats, which can be divided into two types, non-IgG-like and IgG-like, based on their structure with or without Fc region. And there are different BsAb platforms with unique technologies, like knob-in-hole, cross-map, which are well-known designs to avoid heavy chain mispairing and light chain mispairing in asymmetric BsAbs. We'll give more details in part 2. Despite of various BsAb formats, the main mechanisms of BsAb actions are bridging effector cells and target cells, receptor inhibition, receptor activation, and cofactor mimetics. Now there are 13 approved BsAb drugs, most of which are applicated to treat cancer, as well as hematologic and ocular diseases. You can see that most of the drugs are approved in the past 3 years, which represent a rapid development of BsAb in therapy. So if you want to generate your own BsAb, what's the challenges on design and production, and how to overcome them? For non-IgG-like BsAbs and the symmetric BsAbs, the only thing you need to think over is the linker, which may affect the yield and purity. For asymmetric BsAbs, it's more complicated because of chain mispairing. You need appropriate BsAb design to avoid those issues. And for the production part, you may be faced with aggregation, low yield, and purification difficulty to get high purity product, and QC problems to detect the purity. First, let's look at heavy chain mispairing issues. Knob-in-hole and Fc charge pairs are the well-known strategies to avoid heavy chain mispairing. We'll talk more about this. Knob-in-hole involves engineering CH1 domains to create either a hole or knob in each heavy chain to promote Fc heterodimerization. The commonly used variants are V8 and V11. The difference is that V11 has an additional disulfide bond, both of which are highly effective to promote heterodimerization. However, the imbalanced expression of knob and hole still leads to half antibody and homodimer. As you can see in the case, the overexpressed knob or hole exists as half AB and homodimer. We'll address this issue later in the purification part. In addition to knob-in-hole, Fc charge pairs, or electrostatic steering interaction, have also been widely used to avoid heavy chain mispairing. By substituting residuals in two CH3 domains with negatively charged residuals and positively charged residuals respectively, the two heavy chains tend to form heterodimer. And the location of the charge pairs could be CH3 residuals within short distance, or just switching the native charge pairs. So there are different kinds of combination. Here is the case DKK. And similar to knob-in-hole design, the problem of Fc charge pairs is also that the imbalanced expression of two heavy chains could induce half antibody and homodimer. Also, there are some other Fc engineering strategies that could avoid heavy chain mispairing. This is single-chain Fc domain, using a long linker linking two Fc, and the variable region could be byte. The problem for this design is that the yield could be relatively low, and there will be aggregates, we have to do a second purification step to remove aggregates. And the ETYY, the design combined Fc knobbing hole and Fc charge pair. Study. The strand exchange engineered domain is an alternation of human IgG and human IgA sequence in CH3 domain. Now let's move on to light chain mispairing. There are many designs: cross-map, Fab charge pairs, common light chain, Fab knobbing hole, Fab disulfide bonds, single-chain Fab, replacement of CH1Cl with heterodimer, and knobbing hole design. Among these, common light chain is most perfect design for production, but this needs a complex screening step before you get the sequence, so not many people are using this design. In this part, I'll focus on cross-map and Fab charge pairs. The first technology to avoid light chain mispairing is cross-map. There are three designs by interchanging the whole Fab, CH1Cl, or VHVL regions. Cross-map Fab is rarely used because of the side products, and CH1Cl is the most commonly used. Here, please remember to optimize the envelope connecting variable and constant domain, which can make the crossover region better matched in space. Although many scientists have tried cross-map CH1Cl, and there are approved BsAb drugs using cross-map CH1Cl, the results are not always reaching expectation. The two cases shown here have exactly the same backbone, but the variable regions are different. As you can see, the good results only have some half antibody, while the bad one has a list of mispaired byproducts and fragments, which cannot be removed by purification methods. Also, we can try to optimize the plasmid ratio. It's hard to help much. And another design you can try is cross-map VHVL with Fab charged pairs. We get some good case for this design. Fab charge pairs is another recommended technology to avoid light chain mispairing. The first design is 4 charge pairs between 2 variable regions and 2 constant regions on the Fab, and second one combining 3 charge pairs and a group of mutations for Fab knobbing hole design, and the third one is cross-map VHVL on one Fab and Fab charge pair on the other. The case shown here showed correct light chain assembly. There are some other technologies to avoid light chain mispairing: Fab knobbing hole, Fab disulfide bond shift, single-chain Fab, and replacement of CH1Cl with heterodimer. For the Fab knobbing hole design, this can efficiently avoid light chain mispairing. The only problem is that as there is no covalent interaction between the light chain and heavy chain, the SDS page results may look not good. You see some light chain band and 3 to 4 band. And alternative interaction disulfide bonds in fact doesn't work that well. There will be a non-covalent light chain mispairing. And for single-chain Fab, the only problem is that the long linker may lead to some aggregates. And for this, replacement of C1Cl with heterodimer is similar to Fab not being whole, as there is no covalent interaction. The SDS page purity may not look so good. The technologies described above still have some purification problem. Compared to these technologies, in vitro assembly methods may be ideal approaches that could avoid heavy chain mispairing, light chain mispairing, and purification problems at the same time. The first method is Fab arm exchange, or DUBD. Two IgGs with proper mutations on CH3 region are expressed and purified respectively, and then reduced to half antibody and mixed with 1:1 molar ratio, and finally assembled to the right BsAb. The only problem for this method is that the oxidation may take 1 to 2 weeks or even longer. Not being whole in vitro assembly has similar process. Here you can see one knob and hole are expressed and purified separately. You can see half antibody and homodimer. After in vitro assembly, there are only intact target BsAb. One thing to be watched out is using knob-in-hole mutations without additional disulfide bond. Here is a quick summary for asymmetric BsAb designs. First, knob-in-hole and Fc charge pairs are efficient technologies to avoid heavy chain mispairing. However, the imbalanced expression of two heavy chains still lead to half AB and homodimer. Second, Fab charge pairs and Fab knob-in-hole ensure correct light chain assembly, while cross-map and engineered Fab disulfide bonds are not as efficient as expected. And third, in vitro assembly like DUBD knob-in-hole can avoid both heavy chain mispairing and light chain mispairing. Now let's move on to BsAb production and purification strategies. Aggregation is mostly generated from long linker in the design and low pH during purification. Maybe you cannot avoid using long linker, but you can try higher pH in the elution buffer. The yield can be improved by changing expression system, optimizing transfection ratio, and better purification methods. And for purification, there are several strategies to get high purity product, especially for asymmetric BsAbs. To make it easy to remove heavy chain mispaired byproducts, one recommended design is RF mutations. This idea comes from the protein A binding difference between human IgG3 and human IgG1. If the Fc residues histidine 435 and tyrosine 436 was mutated to adenine and phenylalanine, the Fc region cannot bind to protein A anymore. This mutation design is useful for asymmetric BsAb purification. When we introduce these mutations into the overexpressed heavy chain, the half-AB and homodimer in the sample will flow through when loaded to protein A column and will get pure BsAb product. One tip here is please choose protein A resin that does not bend VH domain. And you may ask, what if the RF mutations are not on the overexpressed heavy chain? Don't worry, we have other methods to deal with this situation. In this case, there's no knob-in hole or Fc charge pair on the Fc region, but only RF mutation on one side. So we get two homodimer byproducts, one with RF mutations and one with Y-type Fc. When the sample is loaded to protein A column, the RF homodimer just flows through. The bound target BsAb and byproduct 2 are eluted using pH gradient elution with salt. As you can see, there are two separate peaks. One is our target BsAb, and the other is byproduct 2 we want to remove. Based on these cases, RF mutations are efficient strategy for asymmetric BsAb products and purification. Of course, if there's no RF mutations in the design, you can also choose other purification methods based on the sequence and results. In this case, the red assembly is scFv-Fc with BsAb-Fc, but we also get scFv-Fc monomer and homodimer as byproducts. After analysis of sequence, we find that the byproducts does not bind to protein L, while the target BsAb does. So we use protein L to get high purity BsAb. The principle here is that protein L binds human kappa 1, 3, 4 subtypes, but does not bind kappa 2 and lambda. You can also choose other special affinity columns based on your design and byproducts. And if affinity columns do not work, we'll move on to non-affinity methods. Here comes the case. This is a knob-in hole and cross-map CH1Cl design. The byproducts are half antibody and homodimer. We use IEX to separate the byproducts. You can see the peak corresponding to half antibody and homodimer are perfectly separated. But to be honest, it's not always that good. Sometimes we need to try different buffer and columns. Here is a summary for BsAb production. Different BsAb formats come up with different challenges. For non-IgG-like formats, the first challenge is aggregation. As I mentioned before, if you cannot avoid using long linker, please try to avoid using low pH. The second one is step-free purification. You have to consider if your sequence can bind to any affinity columns like protein A, protein L. If not, we need to do non-affinity purification. The problem is that if the yield is not high, the purity could be low. And for symmetric IgG-like formats, the first challenge is also aggregation, and the second one is low yield. For this one, you can try different expression system or code optimization. And for asymmetric IgG-like formats, you have to consider more. The first one is the chain expression level. As the case is I mentioned before, different chain may have different expression level. You have to optimize the plasmid ratio to get as much as the right assembly of your BsAb, or some easy to remove mispaired byproducts. And for the byproducts, you have to consider what's the byproducts and how to remove it, and also how to detect byproducts. There are multiple analysis methods: SDSPH, CSDS, SEC HPLC, RP-HPLC, HEC-HPLC, and also LC-MS. It would be much easier if you can think it over at the design stage. For example, if the plasmid ratio needs to be adjusted, what byproducts you may get, and if there is a clear purification steps, and what's your necessary QC method to detect byproducts. If you have limited experience on BsAb design and purification, don't worry, Genscript can help. In the last part, please let me introduce Genscript's experience on BsAbs. Genscript has launched its own proprietary TurboCHO platform, which offers higher yield performance within shorter time periods. This cost-effective production system enables small to larger scale antibody production, meeting your requirements at different research stages. So far, Genscript has successfully produced more than 60 different BsAb formats. With the specialized format design, AI-driven code optimization, optimized plasmid ratio, proprietary TurboCHO platform, optimized production process, and multiple purification methods, Genscript can help you to achieve your BsAbs. Here is a BsAb optimization case. The BsAb is 5-SFA Fc design. When it was expressed in ESP293F system, the knob chain is much overexpressed, so the purity is quite low. And because the yield is not high, we cannot move on to a second purification step. So finally, the product doesn't meet needs. And then we did a list of optimization. First, we increased the plasmid ratio of whole, and then express it in our TurboCHO 2.0 system. And when we detect the expression level in the supernatant, we found that whole is overexpressed. So we use CH1 column directly. And finally, we get high purity and much higher yield. This is all we can do for your BsAb expression. And in order to better prove the TurboCHO performance, we specially select some low-yield BsAbs and test them in our TurboCHO system. The result here shows that TurboCHO 2.0 system has increased the yield 2 to 70-fold compared with other commercial systems. And in TurboCHO HP platform, a stable expression system, the expression yield is even higher. As shown in the picture, the yield has increased 20 to 500-fold. If you are looking for expression system for a BsAb, or you have a gram-level antibody expression requirement, our Turbo platform is a good choice. In the end, please allow me to introduce our company, Genscript. Genscript Biotech Corporation is the world's leading technology and service provider of life science R&D and manufacture. Genscript offers research services in 8 areas, including mRNA, oligo, gene editing and molecular biology, peptide synthesis, antibody production, protein expression and purification, and cell line development. If you have any associated requirements, please do not hesitate to contact us. Here is all the content of the bispecific antibody webinar today, and I'm more than happy to answer your questions.
Well, thank you very much, Dr. Chen, for that insightful presentation. So audience members, that does bring us to a Q&A portion of today's webinar. So if you have questions, keep sending them in. We've already received some questions, so let's start off with those. And our first question here for you, Dr. Chen, is what are the advantages and disadvantages of co-expressed BsAb compared with in vitro assembly?
For the advantages of co-expressed BsAbs, I think there are 2 points. One is that lower cost because in vitro assembly will cost about twice for 2 IgG expression and purification. And another advantage is that you can make a stable cell line for co-expressed BsAb and get a much higher branch stability. And for the disadvantage, it is that you may fail to get high purity products if you don't have proper purification methods or if the byproducts are complex. For in vitro assembly, as long as the 2 IgGs are expressed, we can get high purity BsAb. That's it.
Great, thank you, Dr. Chen. And another great question from an audience member. That question is, what is the effect of the choice of purification strategy on the results?
Okay, so here. So the aim of purification is to remove the impurities and to get your target BsAb with high purity. If you don't find a proper purification step, you may fail to separate your BsAb and the byproducts, and you may, or if you may separate them, but it's not so efficient. So the recovery rate of the final yield could be quite low. So as I mentioned in the presentation, it was important to think it over before the production, and maybe additional ARF mutations or adjusting plasmid ratio can help. That's it.
Thank you again. Another interesting question from an audience member. Their question is, what is the long linker and why the existence of long linker would cause BsAb aggregation?
Okay. The long linkers I mentioned include like SF-free linker with about 15 amino acids and BsAb linker with over 30 amino acids. The antibodies with long linker may have long assembly like dimer, trimer, or high molecular weight aggregates. And these aggregates are secreted and exist in the supranatant. And moreover, antibodies with long linker may have lower stability and be more sensitive to low pH, which may aggravate the aggregation.
Thank you for that. Thank you for that comprehensive answer, Dr. Chen. And another question from an audience member, that question is, there are many technologies to avoid heavy chain or light chain mispairing. Can we use them in commercial production? Do they have patent protection?
Yes, most of the technologies, especially for those asymmetric BsAbs, are still protected by patent, except for non-affinity. So if you are making a commercial product, you need to consider, you need to confirm the particular format you want, as if it is in production, or you can contact us for advice.
Great. Thank you again, Dr. Chen. And another question from an audience member. That question is, would you tell us all the purification strategies when evaluating the BsAb projects? Is there any additional charge for using special columns?
Yeah, when we get your sequence, we'll assess what's the format, and if there is obvious mistake, and if there is any advice to optimize the design. You can communicate more with our project manager. They are trained, and also if they can answer your questions, to ask our senior scientists for the help, including me. Yeah.
Great, thank you. Another question from an audience member. The question is, can you help client to design BsAb and avoid mispairing issues?
Yes, we can help with the design, but for the mispairing, we cannot avoid it because, like non-affinity, there is still some heavy chain mispairing. We need to remove it in the purification step. So what we should consider more is how to purify it. That's it.
Thank you. Another great question from an audience member. That question is, what is the fastest turnaround time? If I need a gram level expression, what is your recommended service?
So for now, if you want a small scale antibody expression, the turnaround time could be as short as 1 week. But for bispecific antibody production, as the gene synthesis and the purification is relatively complex, so it should be 2 or 3 weeks. And for gram level expression, QHP, as I mentioned in the presentation, it's enough for several gram level. But if you have a plan for further scale up, like 10 gram, 20 gram, so stable is more recommended.
Thank you. Thank you for that comprehensive answer, Dr. Chen. Another great question from an audience member. That question is, what is CH1 purification?
Okay, in the last case, we use the CH1 column. So maybe you can share my slides so I can point that out.
Sure, Dr. Chen, I can do that for you. Just let me know what slide you want shared.
Okay. The slides, let me check. The slides 31, page 31.
31, okay. Is this the one?
No, oh, we are on different page.
Yes.
No, next, and maybe let me check. Maybe it's 35 or 34.
Okay, so I'll go to 34. This is 34, and then this is 35.
35, yeah. That's
So,
it. Yeah, this one. In this case, we use the CH1 column. Because you can see in the supernatant, I marked here, the whole is overexpressed, and the whole is SCF3 FC. So the byproduct SCF3 FC monomer and homodimer don't have a CH1 region. Only the target bispecific antibody has that region. So we use the column only bound CH1 to purify the target BsAb. Of course, you can also use copper as a special column for this step. One thing you should take care is that the CH1 column may only bound the human CH1 and didn't bound some other, maybe a mouse CH1. And also the kappa column is separated. There are two kinds of kappa column. One is only bound the human kappa, and one only bound the mouse kappa. Just make sure you use the right one. That's it.
great. Thank you, Dr. Chen, for all of those answers. We reached the end of the Q&A portion of this webinar. If you couldn't attend to your questions, not to worry, the team at Genscript may follow up with you later on as well. And attendees, if you have more questions, you can go ahead and jot these emails down. So daniel.kim@genscript.com and then protein@genscript.com. So go ahead and jot those down. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our webinars. Additionally, there's a link in your chat box where you can view that recording. So go ahead and take that down as well. If you have any colleagues, make sure you are sharing that as well. Now, please join us in giving a big thank you to our speaker today, Dr. Xiaoyu Chen, for that fantastic presentation. We hope you found this webinar informative. Have a great day, everyone.
Thank you.